the infectious cycle is another name for what happens to the virus when it gets into a cell. Virus replication is another word that we use to describe that. Okay, I'll use the two words interchangeably. It's the whole thing that happens, all the events that occur when a single particle, a virus particle, attaches to a cell and gets inside, replicates, and releases a lot of particles. And this is all happening in a relatively short time compared to our lifetimes. It can be as short as 20 minutes. It can be a few hours. It can be a few days. So that's the whole sum event of everything that happens, infectious cycle, virus replication. That's what we want to talk about today. So what happens here, the virus gets into the cell. They make multiple copies of the genome. The genomes are packed into particles. And those particles then get out of the cell. So one infecting virus can give rise to hundreds or thousands that come out of the cell and then infect again. So that's the whole infectious cycle. And what I want you to remember at the outset is that all infections, whether it be in a single cell or a plate of cells or in animals, all begin in the same way with the events in a single cell. All right, so today that's what we're going to focus on, what happens in single cells. And I'm going to explore how we use that to understand virus infections. Now here's an example of an infectious cycle. It happens to be the cycle for poliovirus, which is shown up here. And that's because this is a virus I've worked on for over 30 years, so I have a little bias in showing it. But every one is different in many ways, and that's one of the things we'll explore in this course, how the infectious cycles differ for different viruses. And this illustrates what an infectious cycle is. Now, one bit of warning, virologists divide it into steps, so me included, to make it easy to study. And the steps are shown here. We're going to go through them in a moment. But I want you to know, of course, that there are no boundaries in cells. There are no discrete steps that have to happen before something else does in a very specific way. We just do this in order to facilitate its study. So the red arrows are general steps of the replicative cycle, or the infectious cycle, if you will. And the first one is attachment and entry. So all the viruses that we're going to talk about have an obligate step of attaching to a receptor on the cell surface. That's how the viruses get in. There are some examples of viruses that don't have to do that, and we'll get to that when we talk about this specific part of the replication cycle. So the virus's goal, of course, is to get its nucleic acid inside the cell. It's an obligate intracellular parasite. Nucleic acid has to get, in order to do that, it binds to a cell surface receptor. So we have a whole session where we're going to talk about those interactions, but that's the step of the infectious cycle called attachment and entry. The goal is to get the genome in the cell. And in this case, for poliovirus, you see in this picture the capsid is left behind, but in fact the virus capsid does get in, as you will see. And the goal here is to get the nucleic acid, which in the case of polio is an RNA molecule. Now in the textbook, everything is color-coded. So RNAs are green and DNAs are blue, and they're different shades according to whether they're plus or minus. And proteins are thicker bars. So the nucleic acids are squiggly and the proteins are thicker bars. I don't know if that helps anyone, but that's the convention we decided on, at least to make it less confusing. So for poliovirus, the nucleic acid, which is an RNA, has to get into the cell. And this is a mRNA, essentially. It's a plus-strand RNA. And that can be translated immediately as it gets into a cell. So the second step is translation. This is not the case for all viruses. Not all viruses are translation ready as they get in the cell. We'll get to that. But poliovirus can do that. It can translate its mRNA and make viral proteins. And those are shown here on the left side. And some of those viral proteins will eventually go on to make new capsids and make all the new virions that come out of the cell. And other viral proteins will go on to carry out the next step of the infectious cycle, which is genome replication. You have to make more of the viral RNA. And for the case of polio, it does this in a very interesting way that we'll hear more about. It does it on vesicles in the cell, which are these circles here. And the virus replicative process makes more plus-strand RNAs, which are the brighter green color RNAs here. Those RNAs are messenger RNAs, so they can go back and be translated to make more protein, or towards the end of the infectious cycle, they can go on and be incorporated into new virus particles. And again, this is different for different viruses. Some viruses have to make mRNA first. So for example, DNA viruses, when they get in the cell, they can't be translated as DNA. They have to be made into RNA. So this will differ, but this is just a framework to give you an idea of what is happening. So late in infection, and for polio late is about 8 hours after infection, the new proteins and the new RNAs assemble, and that's the assembly step. All viruses have to do that, and they're all very different ways, and we'll look at that in a separate lecture as well. And finally, the virus particles that are made, not just one here, but hundreds or thousands per cell, they have to get out of the cell so that they can spread and infect other cells. That's how they've evolved to do, and that's the step called release. And for poliovirus, the cell actually breaks open, and all the new virus particles are released from the cell. But it's very different for different kinds of viruses, and again, we'll talk about that in detail. Now, this is an example of an infectious cycle that takes place entirely in the cytoplasm of the cell. So here, the nucleus up here is actually an afterthought. You could take the nucleus out of this cell, and poliovirus would not care. It would replicate quite well without a nucleus. And most RNA viruses don't really need the nucleus. There are a few exceptions. Most of the DNA viruses do, because things having to do with DNA, replication and transcription of it, occur in the cell nucleus. So that's an infectious cycle. It has discrete steps that we will talk about in separate lectures. And scientists study viral replication by looking at specific steps in the infectious cycle. Now, before we go on, we have to have some important definitions up front so that you understand what I'm talking about. And these are words that are in the English language and not necessarily have some virus-specific meanings unless you're a virologist. The first is susceptibility or susceptible cell. A susceptible cell means that the cell has a receptor for a virus. That's all it means. Doesn't mean that the virus can replicate in that cell, just that it has a receptor. All right, that is the virologist's definition of a susceptible cell. It has a receptor. A resistant cell has no receptor. It could support virus replication. Maybe if you put the nucleic acid into the cell, by not needing a receptor, the virus could replicate. But that's irrelevant to the definition. Resistance simply means that there's no receptor. A permissive cell can replicate the virus. It may or may not have a receptor. It may or may not be susceptible. All permissive means is that if you put the nucleic acid right in the cytoplasm or nucleus, the virus would replicate. So you see, we're defining very different steps of infection: receptor level steps, susceptibility, and permissivity. A susceptible and permissive cell is the only kind of cell that will take up a virus particle and replicate it. It has to have both in order to support the whole infectious cycle. So those are very specific definitions I want you to remember. We'll be throwing them around all the time throughout this course, and it's important that you know what I'm talking about. If I say susceptible, I'm talking about receptors. And if I mix it up, though, I want you to tell me, because I might sometime or another. All right, so those are our definitions. Now, viruses, if you remember from last time, were discovered around the turn of the 20th century, early 1900s. But for many years, they could not be propagated in cells. I've just told you about the infectious cycle which takes place in a cell. But for the longest time, no one could get viruses to grow in cells reproducibly. So they used animals of all sorts. Mice, hamsters, rabbits, even primates were used. And even in the beginning of the 1900s, there weren't any freezers. So virologists would maintain a virus stock simply by putting it in an animal. And the animal would have a week incubation period. It would die. Take out the organ, grind it up, put it into a new animal, and keep passing it like that, because they couldn't store it, didn't have any freezers. And that was actually an interesting problem, because it turned out that some viruses became amazingly adapted to the wrong animal host. So polio is an example of this. A human virus was passaged from mouse to mouse or monkey to monkey, and it lost all of its original properties for humans. And we'll talk about that later on. So animals were used, and at one point there were freezers and the viruses could be stored. But for a long time, viruses couldn't be grown in animals. Part of the reason for that is not just that we couldn't figure out how to grow cells in culture, but there were no antibiotics, so the cells would get contaminated with bacteria and they would die. So it was not until the 30s that we had antibiotics that we could use for cell culture. Now, in lieu of an animal, some people figured out that one could use an embryonated chicken egg to grow some viruses. And if you take an embryonated chicken egg, it has a little embryo growing in it, and it has a variety of cavities and membranes which serve as the host for different kinds of viruses. So, for example, the embryo is within the amniotic cavity, and you can inject some viruses directly in there, and the virus will grow in the embryo or in the membrane lining the amniotic cavity. You can also inject into the allantoic cavity, which is the larger cavity here, filled with fluid, and also lined with a membrane, chorioallantoic membrane. You can even put viruses in the yolk sac, or for some viruses directly into the embryo. So here is a list of viruses that were at one time grown routinely in embryonated hen eggs or chicken eggs. And we don't really use this anymore because, as you'll see, we have great cell cultures that we can use to grow viruses in. The only exception is influenza virus, which is still grown in the allantoic cavity. And there are two reasons. You get a really good yield of virus. It's very easy to do. And for vaccine production, this is a great way to make millions and millions of doses of vaccine. And so, in fact, the past pandemic season involved the growth of a lot of influenza viruses in embryonated egg, 10 to 12-day-old embryonated eggs. It's all automated now. So actually, when I was a PhD student, I used to work on flu, and I used to inoculate these all by hand. You would drill a hole in the shell and inject the virus through the hole. But now it's all automated because you need millions and millions of eggs to make vaccine. One egg makes about 1 dose of flu vaccine. So, you know, the U.S. alone purchased 150 million doses of vaccine. It's a lot of eggs. So these are all automated systems that pharmaceutical companies have to drill the hole, inject the virus, and incubate it. And then it's a very simple matter of harvesting the fluid, which is full of influenza. You get about 10 mLs per egg, so it's a good yield of influenza virus. But now we don't really do that to study the infectious cycle. You can't really study the infectious cycle in an egg anyway. You need cells in culture. And before 1949, that wasn't really possible, but for animal viruses at least. But in that year, a trio at Children's Hospital in Boston, affiliated with Harvard Medical School, figured out that they could grow poliovirus in human cell culture. So the head of the lab was John Enders, and he had 2 residents working with him, Weller and Robbins. And they had figured out how to make primary cultures from cells. So they got embryos, aborted embryos, and they could take a bit of skin and chop them up, digest them with enzymes, and plate them out in bottles and make cell cultures. And then later on, they figured out how to do this with other kinds of tissues as well. And he actually found that poliovirus would grow in these cultures of embryonic skin. First time polio had been grown in other than an animal, and it was the first time any virus could be grown reproducibly in a cell culture. That was a huge discovery for which he got the Nobel Prize in 1954. And the story goes that John Enders got a call from the Nobel Committee, and they said, "We want to give you the Nobel Prize." And he said, "No, you have to also give it to Weller and Robbins because they helped me do this." And they did, which is very unusual. So the 3 of them got the Nobel Prize, and he made the cover of Time magazine in 1961. Virologist John Enders. This was a really big deal. So this ushered in modern virology. It ushered in vaccines. So Jonas Salk was able to make a polio vaccine because of that discovery, growing viruses in cells. And from that point on, virology exploded. So nowadays we use cell cultures routinely to study the infectious cycle of viruses. And they let us do many things that cannot be done in an animal. And there are 3 general kinds of cell cultures that we use to grow viruses in. The first is very similar to what John Enders used. They're called primary cell cultures. You take a tissue from an embryo of some kind, even a chicken embryo. Foreskins are very easy to get, especially in medical centers. You just mince up the tissue. you digest it with an enzyme to make single cells, and then you plate those onto a plastic culture dish and cover it with liquid medium. So this is an example of primary human foreskin fibroblasts. And these will grow and fill the plate nicely. You can then remove the cells from the plate with trypsin, dilute them, and place them on a fresh plate, which is a process called cell passage. And you can grow these cells for 10 to 20, maybe 30 passages at the most, and then they die. They senesce. The ends of their chromosomes go away because they're not making telomerase, and they die. And that was, of course, a limitation early on until people figured out how to get cells to grow longer, and that's where we have these continuous cell lines. And these are two examples. This is a mouse fibroblast cell line made from embryos. And here is a human epithelial cell line called HeLa cells, which is from a cervical carcinoma. And these cells live forever. They are immortal because they have been transformed. Either spontaneously or by a tumor virus of some sort or by other means, so that they keep the ends of their chromosomes the right length by producing telomerase. And you can grow these forever in the laboratory. So this is what many virologists use. Now the problem with these cells is that they're abnormal. They have odd numbers of chromosomes. They have more than the usual number. And often each cell has a different number of chromosomes from the other. So we call that aneuploid. And so they're okay for certain kinds of experiments, but for other purposes you want a more normal cell line that you can grow for an extended period of time, and that's where diploid cell strains come into play. The first one made was a famous one called WI38, made from human embryonic lung. And these will grow much longer than primary fibroblasts, maybe 100 passages. They have a diploid number of chromosomes, so they're normal, morphologically normal, they're not transformed, but they do have a finite lifespan. So you keep a lot in your freezer, and when they start to die out, you throw them out and replace them. Now, the way we grow cells in the lab is in an incubator, of course, at 37 degrees. The incubators are humidified with 5% carbon dioxide to buffer the medium, because as the cells grow, they excrete acid into the medium, and that wouldn't be good if it weren't buffered. And we use plastic nowadays. In the old days, they used to use glass and wash them and reuse them. But now we have a petroleum industry that supplies us with plastic of various kinds. We have flasks, which are down here, which you use to maintain cell lines, and plates where we do virus experiments in them. So that in each case, there's a flat surface of the plate, and they're different sizes. It's covered with a medium which provides nutrients. It has serum in it and a buffer. And it also has phenol red, which is a color indicator. When they get acidic, you can tell when they turn yellow, or if they become alkaline, they become redder. We can also grow cells in what we call suspension cultures. So the previous picture was of an incubator full of plates and flasks where the cells are growing in a monolayer on the plastic surface, and that is where we do most of our virus experiments. But sometimes you need more cells than you can grow on the plastic plates. And for that case, in the case of HeLa cells anyway, we can grow the cells in what we call suspension cultures. And here we take a bottle which is full of cells, and there is a magnetic spinner there hung from a glass rod, and it's sitting on top of a magnetic stir plate. And in that stir plate there is a magnet driven by a motor which drives the spinner inside the flask. So this is constantly stirring at a low speed, and it's in a warm room. And so the cells multiply very nicely, and you can get up to 400,000, 500,000 cells per milliliter. So that's a lot of cells, and when we need to do big experiments, we grow this. Now these are HeLa cells, which we and many, many other laboratories use extensively. And about a year ago, I wrote a post about HeLa cells, which were derived from a woman named Henrietta Lacks. And in this post, I described where the cells came from. So she was a woman who had a cervical tumor in 1951. And a piece of it was taken out at Johns Hopkins, and this piece of tumor gave rise to HeLa cells. So this fellow, George Gay, was trying to make cell lines that would be immortal, that would live forever. Nobody had been able to do it up until this time. And he found in her cells a cell line that would do that. Now, she died in October of '51, and he went on television that day, and he had a vial of cells in his hands, and he said that he'd made a line of cells that propagated like any other, and they're called HeLa cells because of the first two letters of her first and last names. So after I wrote this post, I got an email from an author, Rebecca Skloot, who was writing a book about HeLa cells. And we started corresponding, and it turned out she is publishing this book next week. It's called The Immortal Life of Henrietta Lacks. And it's here on the cover. It says, "Doctors took her cells without asking. Those cells never died. They launched a medical revolution and a multi-million-dollar industry. More than 20 years later, her children found out their lives would never be the same." So actually, the family didn't know that these cells were from her. So this book is the story of that. So I read it, actually, to make sure she got the science right. So if you read it, the science will be correct. But the neat thing is that she's coming here next week to give a seminar or a talk. I shouldn't call it a seminar. It's not a science seminar. It's the kind of talks that writers give. She's going to read a bit from her book and tell some stories about the family. And so that's over in Fairchild 700 at noon next week. So that should be interesting. So anyway, getting back to virology, how do you know that a virus is growing in a cell? Well, John Enders, when he took his primary skin fibroblasts and he infected them with poliovirus, he knew because the cells were killed by virus infection. And when a virus kills cells or makes changes in it, we call that cytopathic effect, or CPE. So we're guilty of making a lot of acronyms, but this is the one I will use a lot in this course, CPE. And it simply means the changes induced in cells as viruses replicate in them. So here's an example of virus-induced cytopathic effect. These happen to be mouse fibroblasts, which have been infected with poliovirus. And up at the upper left is the monolayer of cells growing in the plastic dish before virus infection. And this is 4, 8, and 12 hours after infection. So you see, even at 4 hours after infection, it looks different. And I can tell you what's happening here is that a lot of cells are starting to round up. They're infected, they're not feeling well, and they're beginning to detach from the dish. And that's more extensive, obviously. At 8 hours, most of the cells are floating. There are a few attached still. And then by 12 hours, many of them have broken. They're no longer host cells present. They're all clumped up, and there's no cell left on the plate. So this is what John Enders saw, and that's how he knew the virus was growing. And in those days, there weren't many better ways to assay for virus replication. The other thing that John Enders did was to take the virus that he grew and he put it in a mouse, and it paralyzed the mouse. So he knew he had grown poliovirus. But you can't do that with every virus, and that was the problem at the time. Now, before we go on, he was very lucky that he picked polio to do this, because there are many, many viruses that will not cause any visible changes in cells as they infect them. They are non-cytopathic viruses. So if he had picked one of those, he would have looked at his cells and said, it doesn't grow, and that would have been the end of it. Okay, so not every virus is cytopathic. Not every virus causes such visible changes. You can detect viral replication in other ways, and we'll now move on to talk about how that can be done. But I want you to know that for many of the viruses that we talk about, they don't kill cells like this, yet they still can cause disease. And that's an interesting conundrum that we'll get back to. Now, killing cells like that by making them round up and come off the plate isn't the only way that a virus can cause changes in cells. Another way is by the formation of what we call syncytia. It's the plural for syncytium. And a syncytium is what results when cells fuse. And you get a big cell like this one full of nuclei. So it's a multinucleate cell, essentially. It's called the syncytium. So many viruses do this, and we're not yet at the point in this course where we can understand how this happens, but essentially when certain viruses infect cells, they display viral proteins on the cell surface which cause the cell to fuse with a neighboring cell. Well, this will be perfectly clear in a few weeks. And these virus-infected cells fuse, and then in a plate, of course, where there is a continuous monolayer of cells, you can have many cells fusing, and you make a syncytium. So here's what one really looks like under the microscope. So these are light microscope images that I'm showing you. Here's some virus-infected cells. Here's a syncytium. It has a bunch of nuclei in it. One giant cell with many nuclei. And this can actually be used to diagnose some virus infections, at least in the old days. If you were suspected of having measles, this would be one way to find out very quickly if you had measles, because that was one of the few viruses known that would cause this kind of cytopathic effect. So a syncytium formation is another kind of cytopathic effect. And here's a list. Yes?
You have to use an electron microscope, though. Isn't that very expensive?
No, that's just a, this is not an electron. This is just a light microscope.
Oh.
So that's easy to do in the lab. Yeah. Yeah, these are very low magnification images, sure. Sorry, say that again?
Where's the boundary of that cell?
Oh, the boundary of the syncytium is right here. It's right there. Here, there. And there's another one here, which happens to have 2 floating cells on it. And there's a very big one here, I think, which here and has cells on top of it. Okay. Yes?
When you form a syncytium, do you generally lose the circular shape, or will it maintain the same basic cellular shape, just be larger and multinucleated?
It depends. Some of them they have triangulated or cuboidal shapes like this. Others are more circular. It depends on the cell and the virus. There's no rule. Yes?
And the syncytium will still adhere to the plate?
It will for a while, and eventually. So most of the viruses that make syncytia are produced by a process called budding, where they come off the surface of the cell. So they don't kill the cell initially, but after, say, hours or days of that process, they will eventually break up and come off the plate. Yeah. But you can see in these are attached to the plate. This is an early stage in syncytium formation. So these are just some examples of different kinds of cytopathic effects that different viruses produce, just to show you the range that can be observed. There are, on the one hand, what we call morphological alterations, which include syncytium formation, as we've just seen, the changes in the nucleus, induction of vacuoles in the cytoplasm, the movement of the chromosomes in the nucleus, margination, meaning they move to the nuclear envelope, breaking up of chromosomes, detachment of cells, rounding up, which I've shown you also. And these are characteristic of many different viruses. And then another set of cytopathic effects are what we call inclusion bodies, that if you look inside the cell at high enough magnification, you can see accumulations of different things. Yes?
Do all cytopathic effects ultimately kill the cells?
Yes, they all do. And how long they take differs, but eventually they will all kill the cell. So here we have inclusion bodies of different sorts. And in the old days, again, these used to be diagnostic for certain virus infections. So virions in the nucleus for adenoviruses, negri bodies, which are virions in the cytoplasm, very characteristic of rabies virus. So again, if someone was suspected of having rabies, one could look at cells from the person, and these negri bodies, which stained in a very specific way, would be a telltale sign that was a rabies-infected cell. Factories in the cytoplasm called Guarnieri bodies, where viruses are assembling with pox viruses. Some viruses put clumps of ribosomes in the virions or clumps of chromatin in the nucleus. So a whole range of things can happen. We understand some of these, but not all of them by any means. All right, so John Enders was stuck with assaying poliovirus by looking at how it killed cells and by taking his virus and putting it in animals. But those were crude methods, and they rapidly evolved into what we have today, the assay of viruses. And I want to spend some time talking about this. 2 distinct ways of assaying viruses. First, the measurement of infectious units, infectious virus particles, and the measurement of particles and their components. So these are 2 very distinct things. So an infectious virus is a particle, but not every particle is infectious. So very different things are measured here. And the first that I want to talk about is the plaque assay. And this is one that permeates all of virology, which began in the 1930s. The virologists who studied bacteriophages, the viruses that infect bacteria, developed the plaque assay to study the multiplication of phages in their host. And here's a photograph of a plate. This is an agar plate, which is used to grow bacteria. So it has a thick layer of agar. And what was done here was to mix bacteria, a culture of bacteria, a liquid culture, with a phage preparation, and then put it on top, spread it over the top of the agar. When you put that in the incubator, the bacteria form what we call a lawn. And probably many of you have done this in a lab somewhere at some time. It's a very popular lab experiment to do. And as the bacteria grow in, the phages are killing them and making holes in the lawn. And you can count the number of holes, or plaques as we call them, and develop a titer, as you'll see in a moment, for the virus stock that you have. So this was done starting in the 30s. The bacteriophage scientists didn't have to have cell cultures because they grew their viruses and bacteria. So they didn't have the problem that John Enders solved. But in 1952, Renato Dulbecco, who was a phage virologist and had been doing plaque assays, he said, this has to work for animal viruses. So by 1952, we had John Enders showing that viruses could grow in cells. We had HeLa cells developed. So he tried it, and he got it to work. He developed a plaque assay for animal viruses. And he did get the Nobel Prize in 1975 for that and other work that he had done with tumor viruses. And here's, today I just pulled up this paper, which is from April 1952. Production of plaques in monolayer tissue cultures by single particles of an animal virus. And I like the first sentence. Research on the growth characteristics and genetic properties of animal viruses has stood greatly in need of improved quantitative techniques, such as those used in the related field of bacteriophage studies. So basically, he's saying we need to be able to quantitate viruses like we can do phages. So he had tried this, and he got it to work. So how do you do a plaque assay? You start by making dilutions, serial dilutions of virus, and typically we make 10-fold serial dilution. So you start with a virus stock in a tube. You take, in this case, 0.1 ml, and you put it into 0.9 ml of buffer. That's a 10-fold dilution. And you keep doing that serially as long as you need to go to get a good endpoint, as you'll see. For many viruses, you need to go out to 10 to the -7th. Then you take a little bit of each dilution and you put it on a monolayer of cells. So now you've grown in your incubator a bunch of small plates which have confluent monolayers of, say, HeLa cells or some other cell type. And then on the day you're going to do the plaque assay, you remove the medium and you add your virus dilution to the surface. And here we're adding 0.1 ml, and that adds another 10-fold dilution to the calculation. So you add the virus and you let it absorb to the cell. And we do that for about an hour. We let the virus attach to the receptors on the cell surface. And then we cover the cells with a semi-solid overlay, or medium, which will restrict the diffusion of the virus. If we put liquid overlay on these cells, they would all die, as we saw in that slide earlier. They would slowly develop cytopathic effect and die, and you wouldn't be able to quantitate anything. But if you put a semi-solid overlay on, you restrict the spread of the virus, and it makes, instead of spreading throughout the whole culture, they make small holes in the cell monolayer. So you incubate it for the amount of time, and the virus destruction makes little holes, which are plaques, just like the bacteriophage plaques. And you can count the number of plaques. You can remove the overlay, stain the cells with a nice dye, count them, and you can calculate the titer. So here, for example, on this plate we have 17 plaques. And that gives you a titer of 1.7 times 10 to the 8th plaque-forming units per ml, PFU per ml, because that's what we're measuring, a plaque-forming unit. At this point, we can't say that this is an infectious unit or a single virus. A plaque-forming unit is the correct way to call that. So let's look in a little up close at how a plaque forms. Here's our plate of cells, and when we add our virus, it gets into cells initially, and now here we have one cell in which the virus is multiplying. It's red. So this is a magnification of a very small part of this monolayer. As the virus is produced in that cell, it spreads to neighboring cells. And again, the spread is restricted by the agar overlay. If we had liquid, the virus would diffuse throughout the whole culture and kill all the cells. Here, the spread is restricted by the agar. So that's a very important component of the plaque assay. And that focus of infected cells gradually grows larger until it's big enough to see with the naked eye. Then you stop the experiment, you stain the cells. So here is a high magnification image of a plaque produced by a herpes virus in cells. So here's a single plaque. So we're zooming in. These are all single cells, and you can see the monolayer around is quite nice. But here where the plaque is, the cells have mostly detached, and in the middle they're all rounded up and clumped up. So if you stained this with a dye, all these cells would fall off and you'd have a hole in the monolayer. Now this virus used to do this plaque assay happened to have a gene that was put into it that allows you to detect the color in the infected cells. And this monolayer was stained with a dye that reacts and gives you a blue color when the virus is present. So it's the same plaque except it's stained, and you can see the presence of virus in all the cells. This is a plaque assay of influenza virus, and this shows you that, again, multiple plates inoculated with 10-fold serial dilutions. The plaques are allowed to grow. They're nice size. At some point you stain them. In this case, the dye is called crystal violet. It stains all the living cells, and where the cells were killed, there's a hole in the monolayer, and you can count the number of cells. Now, this illustrates the point that you want to have enough plaques on a plate to be able to calculate your titer. Let's say you used a dilution of this sort where you have one plaque here. Now, if I plated out 0.1 ml onto this plate, that would mean that the virus titer would, in theory, the virus stock would have a titer of 10 plaque-forming units per mil. But you can see in the duplicate plate, there's no plaque. So counting one plaque is too error-prone. You have to have usually between 10 and 100 plaques to get a good titer. Okay, so I would count either this one or this one to develop the titer here. So now we can assay viruses by plaque assay. It's a beautiful assay. But the key question, of course, is how many viruses form a plaque? Because if you want to say that each plaque starts from a virus so that you can get an idea of how many viruses are in the culture, you need to answer that question. To do that, what you do is a dose-response curve. Very simple experiment. You make dilutions of virus again. You don't do 10-fold dilutions. You do much smaller dilutions. And you simply plate those dilutions on your cells. You do a plaque assay. If one infectious virus will form a plaque, then the dose-response curve will be a line. That's what we call one-hit kinetics. One virus is enough to make a plaque. So in mathematical analysis of one-hit kinetics, the number of plaques is directly proportional to the first power of the concentration of virus inoculated. Simply put, it's a straight line. The straight line between the relative concentration of virus and the number of plaques. So that proved, and this was actually done by Delbecco in his first paper, to show that one plaque originates from a virus, a single virus. There are some viruses for which you need two particles to make a plaque. So what does the curve look like? For those, two-hit kinetics, you need two particles to make a plaque. The number of plaques is proportional to the square of the concentration. So you get a line, a curve, such as this one. And some viruses, you need two particles to make a plaque because the genome is in two pieces. One piece is in one virus and the other is in a separate, so they both have to infect cells in order to make a plaque. So this was very important because it showed that single viruses could make plaques, and that led to genetic analysis of viruses because it became possible to do what we call plaque purification and isolate pure populations of viruses. So you could take a clinical isolate from an animal or a human and plaque it in cells and get very genetically pure populations. This is called plaque purification, and it's a method for producing clonal virus stocks. You usually do this 3 times because there can be 2 viruses which by chance fall into the same area and make what looks like a single plaque, but it's in fact 2. So to reduce that possibility, you do it 3 times. You have an agar overlay. You can see the plaques typically without staining them. You take a plastic pipette, you plunge it into the agar over the plaque, and you put that plug of agar into buffer, and now the virus comes out, and you can use it, grow it up, and you have a plaque purified stock. So that's an important technique for viral genetics. Now, not all viruses will form plaques. Many, many don't. So you have to have other ways to assay them. And I want to go through a few of those. One of them is called the endpoint dilution assay. These are for viruses that will kill cells. They will form cytopathic effects, but they don't form a plaque. So what you do here is you take a 96-well plate or a similar cell culture vessel. Each well here has got a flat bottom, and you put cells in it, and you make a monolayer. and then you infect each row of wells with a different dilution of your virus stock. So all these wells get 10 to the minus 2 dilution, all these get 10 to the minus 3, etc. Then you incubate your cells, and you look for the ones that are killed. You look for the cells, or the wells, where there's obvious cytopathic effect. And you can do that by staining the monolayers with that blue dye, and you put a plus when you see cytopathic effect and a minus when you don't. You can see the trend is, as you dilute the virus, fewer wells have cytopathic effect. And then you look for the dilution where half of the wells develop cytopathic effect. So in this example, it happens to be minus 5. There are 5 positive wells. And that's because I made up this example, because in real life it never falls on a single dilution, and you have to use interpolation methods to figure out. It's usually between 2 dilutions, unless you're very lucky. And what this does is give you what's called a TCID50, tissue culture infectious dose 50%. That is the amount of virus that will infect, or in this case cause CPE in 50% of the wells. In this case, 10 to the 5. This virus stock has 10 to the 5 TCID50s per mil. So this can be used for viruses that don't form plaques, and it's still very much in use today. Another issue we need to deal with when we talk about viruses and plaques is the particle to PFU ratio. Now this is just a number, which is a ratio of the number of virus particles in a sample divided by the number of infectious particles. So how do we get this? Well, the bottom number is easy. You can do a plaque assay. The top number, you have to actually know physically how many virus particles there are. That's harder to do. You could look in an electron microscope. You can do spectrophotometric measurements, some kind of physical measurement to get the number of particles. And then you take the ratio. And surprisingly, this is not 1 for all viruses. You might think it would be, but it's not. For bacteriophages, for many of them, it's 1. But for many animal viruses, it's 10 or 100 or even thousands. And this is a problem when we study viruses, because if you have a virus that you're studying where the particle to PFU ratio is, say, 10,000, so say when you infect a cell with virus, you're putting a lot of non-infectious particles in the cell. So if you're doing biochemical studies or visual studies of the infected cell, you might not know if what you're seeing is a consequence of the infectious or the non-infectious part of the population. So this has to be kept in mind whenever you do virology with some of these viruses with high ratios. So these are just an example of some of the ratios of different viruses. This one, Semliki forest virus, is a well-behaved virus. It has a pretty good ratio, 1 to 2. Most or at least half of the particles are infectious. But look at these numbers for other viruses. 200 to 10,000 for papillomaviruses. So for every infectious particle, there are 10,000 ones that are not infectious. So that's a problem that you have to keep in mind. Now, how does this correlate with what I told you about the dose-response curve? If you're a little confused, the linear nature of that curve, remember, showed that a single particle can initiate infection. Can, but may not always initiate infection. The high particle to PFU ratio shows that not all viruses are successful at initiating an infection. So that's how you can rationalize the 2 apparent paradoxes. Now, why aren't all particles infectious? In fact, we don't really know for most viruses. We have a bunch of ideas. Some of them may be damaged. You grow them in cells in the lab, they get damaged, so they're not infectious. Some of them may have mutations in them, which arise during replication. Or a third possibility is that, remember, the replication cycle of a virus, you have a lot of steps that have to happen, and if you mess up at one step, the whole cycle is gone. So that could be part of the problem too. If there's an error at any step in the cycle, you don't get infectivity. We really don't understand this fully up to this day, but it's something you need to keep in mind with virology. Now, the replication cycle, the way we study it is by using a technique called the one-step growth cycle. And this was first done in 1939. 2 virologists, Ellis and Delbruck, worked this out for bacteriophages. So what they did, they add virus to cells. So they have bacteria growing in a liquid broth. They take a little bit of that broth, they add virus to it, and let the virus attach. And they let it attach only long enough that the virus needs, which they figured out. And then they dilute the culture. And that prevents more viruses from attaching. So the attachment of viruses to receptors is concentration dependent. When you dilute, you stop absorption. So the effect is to synchronize the infection. So all the infection that's going to happen in that culture is now finished. There's not going to be any more new infections. And then what they did is take a sample of the culture every few minutes. They put the infected culture in an incubator, take a sample every few minutes, and look for infectious bacteriophages. And they found something which to them was very surprising. On the upper left, what they found was this curve here. So this is time and the amount of infectious viruses produced. And they use a plaque assay to do these studies. So what they found first is there was an initial lag period. No virus could be detected, or very little virus in these initial time points. And then there was a rapid increase in virus production, and finally a plateau. And they called this a burst. And the burst, you'll see in the literature for many years, the burst experiments reflect this very rapid increase in virus production. When you have many cells infected, or all the cells, this is the pattern that you observe. They are all synchronously infected. The virus gets in, multiplies, and you have a burst all at once. If you put less virus into the culture, you get this kind of infection. You initially get a burst, but not all the cells are making virus. So the virus that's released goes on and infects other cells, takes longer because it's a dilute culture, and then you get a second burst. This little phase in the beginning puzzled them for a while until they figured out that during this period, which they called the eclipse, what happens is the virus has gotten into the cell, or it's placed its nucleic acid in the cell, and it's no longer infectious. So you can't detect infectivity because the nucleic acid is naked in the cell, it's replicating, and it doesn't score as infectious in this assay. Here, at this point now, where you start to see plaques being produced or infectious viruses being produced, that is when the newly assembled viruses are formed. So that's an important concept, this eclipse period. And finally, what they did was experiments where they didn't just look at the total virus produced in the culture, but they separated the bacteria from the supernatant. And what they found was that you can find new infectious viruses in the bacteria before they come out into the medium. So from this and other experiments came the notion that the virus gets in the cell, it makes building blocks, they assemble, and then they're released. So you can see the assembled viruses inside the cell before you see them in the medium. Now, of course, this was rapidly adopted for animal viruses, and this is an example of a one-step growth curve for adenovirus. One step, again, because all the cells are infected, and they're going through the infectious cycle, all of those steps, attachment, uncoating, replication, and so forth, all at the same time. So with adenovirus, you infect cells in culture, and what we've done here is look at the time after adsorption or adding the virus and the amount of virus produced in two ways. We're looking at intracellular virus, the virus just inside the cell, and we're looking at virus that's in the medium of the cell. So you separate the two. What you find is an eclipse period, just like with the bacteriophages, and then virus production inside the cell begins, and eventually it plateaus when the cells die. If you look at the extra, yes?
Why is there a slight decrease?
That's the eclipse phase. In fact, you attach viruses to the cell, and so when they're sitting on the cell surface, you can detect their infectivity. And then when you start the experiment, you put the cells in the incubator, the nucleic acid gets out of the virion, and so that's the decrease in infectivity. So this can be actually much more prominent depending on how much virus you start with. For some, you could see that zero time point is way up here, it drops all the way down, and that's reflective of the nucleic acid coming out of the virus particle. Now, if you compare that pattern with the extracellular virus production, what you see is that you don't see viruses in the medium until 16 hours and beyond. So in this period here, between 12 and 16 hours, you see infectious viruses in the cell, but they're not yet put out of the cell. They're still inside the cytoplasm or the nucleus, wherever they may be. Then they are released into the medium and they accumulate there. So not only is there an eclipse period during which the nucleic acid is not infectious, there's what's called a latent period. That is just the time before you see infectious viruses in the extracellular fluid. Okay, so that was a typical one-step growth curve. We can use this to study all aspects of viruses, and we'll see in the duration of this course how we use this in many, many different ways. And this is just to emphasize that, remember, one virus gets into a cell, makes the building blocks, and many, many new the viruses are assembled, which then leave the cell. And that's in contrast to bacteria. If you inoculate a culture with bacteria, it begins to multiply immediately. So you see an increase immediately. There's no lag period. There's no eclipse. The bacteria start dividing. And this is fundamentally different from how the viruses multiply. Now, this may seem obvious to you today, but to the virologists who were studying this in the early days, they were surprised that viruses didn't multiply like this. And it was left to them to figure out why, and I've told you that. So synchronous infection is really important for a one-step growth cycle. And again, you do that by putting enough virus on cells and diluting the infection afterwards. So how do you know how much virus you're adding to cells? That's a key question. And that depends on a parameter called multiplicity of infection, or MOI. It's the number of infectious particles that you add per cell. Very simple. So if you have 1 million cells and you want to add 10 particles per cell, the MOI is 10. It is not the same as the number of infectious particles each cell receives. So if I add 10 to the 7th virions to 1 million cells, that's an MOI of 10, but each cell does not receive 10 virions. So if there are 54 students in this class and I have a bucket of 54 tennis balls and I throw it at you, you're not each going to get one tennis ball. It's the same thing with viruses. It depends on chance, assuming that I can distribute it as well as I can. Some will get none, some will get 1, maybe 2 and 3. And guess what? That's described by a mathematical equation called the Poisson distribution. So because infection depends on random collision of virus and cells, just like if I throw tennis balls at you, it's a random collision. When you mix virus with cells, some get 1, 2, or 3 or more particles. Tennis balls, virus particles, it's the same thing. And that's determined, the distribution per cell is determined by the Poisson distribution. So there is a formula associated with this right here. This is the formula for the Poisson distribution. And for viruses, it's quite simple, actually. pK is the fraction of cells infected by K virus particles. Okay, so that's what fraction gets K virus particles. M is the multiplicity of infection, how many viruses you add per cell. And for 0, you can shorten this whole equation to this. So the fraction of cells, of uninfected cells, those getting 0 particles, is e to the minus m, where m is the multiplicity of infection. So you can calculate very readily cells that are uninfected, cells that get 1 particle, or cells that are multiply infected. So here, this is the sum of all this is the sum of the probabilities of 0 and P1 here subtracted from 1. So let's look at some examples of this. If you take 1 million cells and infect them at an MOI of 10, you plug those numbers into the equation, you get 45 uninfected cells, you get 450 cells with 1 particle, and the rest get more than 1. So again, not everybody gets 10. It's determined by this distribution. If you infect at an MOI of 1, 37% of the cells are uninfected, 37% get 1 particle, and 26% receive more than 1 particle. So again, you would think intuitively they should all get 1, but it's not the case. And finally, if you go way down, 0.001, 99% of the cells are infected, and very few receive 1 particle, and hardly any receive more than 1 particle. So sometimes you do want to do experiments under these conditions if you want to look at multiple cycles of infection, because here very few cells are initially infected. So if you want to see 2 or 3 infectious cycles, you can do this. So in the course of these lectures, you'll see different ways that this is manipulated to study virus multiplication. Now, there are other methods that we can use to study viruses, other physical methods, including hemagglutination. Electron microscopy is simply a very powerful microscope. We can measure viral enzymes. There are immunologically based methods, and there are nucleic acid-based methods. This is an example of one physical method. It's called hemagglutination. And it uses red blood cells. And it was developed in 1941 here in New York by George Hurst, the first rapid assay for viruses, but not a plaque assay, just a physical assay for viruses. And it's based on the property that many viruses can bind to red blood cells. So in fact, their receptor that they use to attach to cells is also present on red blood cells. And it's very often used for influenza virus, which binds to sialic acid, a sugar. When you add virus to certain kinds of red blood cells, has to be a certain species, the virus will stick to the red blood cells, and then the red blood cells will stick to each other, and they form a lattice, and that's hemagglutination. So you do this in a 96-well plate using wells that are curved. So if you just added red blood cells to these wells, they'd form a nice button at the bottom in about 20 minutes. But if you have viruses in there, they can't form a button because they make a lattice, and you get nice coating like this one here. So what you do is you make two-fold dilutions of virus, you mix them with red blood cells, and you incubate them, and you see where the hemagglutination stops. So you can see B and C don't have any virus in them. They're not hemagglutinating at all. And let's find one that's a good hemaggglutinator. Here's good. D is a good one. It hemaggglutinates out to, let's say, 1024. So that would be the end point. So we would say that virus has an HA titer of 1024. And this way you can very quickly get relative physical amounts of viruses in different preparations. And a variant of this is still used to this day. Yes?
Are you just using a visual?
Yes. Yeah, you just look at it. You put it on a light box, and you can readily say 1024 or nothing or 32. It's a very nice assay. Yeah. I mean, you can imagine it could be automated, but it hasn't been done. You can measure viral enzymes. Many virus particles have nucleic acid polymerase to replicate their genome, whether it be RNA or DNA, and that can be readily assayed. So the viruses that can be assayed in this way are typically enveloped. So you can add a little bit of detergent to the virus particles to permeabilize the envelope, and then you add a nucleic acid precursor like ATP or CTP labeled with something that can be detected, radioactivity or some other kind of label. And you can then detect the incorporation of the precursor, the triphosphate, into DNA or RNA. There are many, many methods for doing this, and they're most often done with retroviruses. And a lot of retros don't transform cells, which is another way you could detect virus presence, which we will talk about, and they don't form plaques. So you'll see in many, many papers them doing what's called reverse transcriptase or RT assays. They're simply measuring the amount of virus in the particle. So here's an experiment where 3 different kinds of cells were infected with this virus called XMRV. This is a new virus that's just been discovered in the last couple of years. It's implicated in prostate cancer, and it's implicated in chronic fatigue syndrome. So what they did in this experiment is to infect 3 different cell lines, mock infected, and then at different days after infection, they take a little bit of the cell culture medium and do a reverse transcriptase assay. Look for incorporation of a radioactive precursor into nucleic acid. And then they spot the product on this filter and expose it to X-ray film. And you can see here very nicely XMRV in this cell at about 2 to 3 days you see the production of reverse transcriptase. Now this is not an infectious assay, but it tells you that particles are being produced. And it's somewhat comparative. You can see this, it grows better in this cell than in this, and it doesn't grow at all in the DU145 cell. So that's an assay you will often see used for retroviruses. There are many antibody-based assays that can be used to detect viral antigens in cells. So here you're looking for a specific viral protein, and if you have an antibody to it, you could label the antibody with a fluorescent indicator, or you could use a second antibody which is also labeled, and that second antibody is directed against the first. And you can put these antibodies onto cells in culture. And here's an example of cells that are infected with a herpes virus, and they are stained with an antibody to a viral protein, and that's the yellow labeled antibody in the nucleus of the cell. And the cell has been stained with an antibody to a cellular protein to mark the cytoplasm. So this is done very frequently. We also use antibodies to detect viral proteins. And by Western blot analysis, you fractionate, say, viruses or virus-infected cells by gel electrophoresis. The proteins can be transferred to a nitrocellulose sheet, and then if you add the antibodies to that sheet, they will bind to the proteins, and you can then detect the antibodies by your favorite detection methods. This can be used for diagnosis. In the early days of AIDS, it was used to diagnose AIDS infections. And it's used, as you might imagine, extensively in the lab to look at viral proteins in cells and their interaction with cellular proteins. Nowadays, many viral infections are diagnosed by ELISA, enzyme-linked immunosorbent assays. You can look for either a viral protein or an antibody to the protein. So if you're looking for a viral protein, so nowadays to diagnose AIDS, you would look for one of the viral proteins in blood. So you put an antibody on a solid support, which is directed against the antigen you're looking for. This is called the capture antibody. This can be a plastic dish. You then add the serum sample, and if there's viral antigen in it, it will bind to that antibody. Then you add a second antibody to the protein, which has an indicator on it, and then you can do immunofluorescence. And this is a variant of the same technique, except here we're looking for antibodies in someone's serum. So for example, if you want to know if someone has been infected with a virus, you look for antibodies. You immobilize the viral protein on the plastic, so you have lots of this that you can put on the plastic. You pass the serum over. If there are antibodies, it will bind to the antigen, and then you detect it with a second antibody. And in many, many experiments now, we use fluorescent proteins of different color. The first, of course, green fluorescent protein from this jellyfish, which is a lovely protein. It looks like a basket with a light bulb in it. The active site, of course, is in the center. And this is famous here, of course, because Marty Chalfie was the first to put it in a living organism. And you can see it without fixing, without substrates, coenzymes, anything. It's beautiful. And it turns out that many viral proteins work when they're fused to GFP or all the derivatives, the different colors of GFP that are available. And you can track all steps of a virus infection by using this. So here's an example, two examples. Here's a herpes virus where GFP has been placed in the viral genome. When you infect a neuron in culture with this herpes virus, the virus produces GFP, and you can see the cell body and all the neurites have GFP. So you can actually use this to track infections in an animal. And this is an example where HIV is used. The GFP is placed into the genome in such a way that the particles are now labeled green. So these are individual virus particles. These are infecting a cell. The cell is labeled with antibodies to tubulin. And so this shows that the virus actually moves towards the middle of the cell by surfing along these tubulin molecules. So many, many fascinating experiments you can do with these viruses. And you can do real-time labeling. Here's a neuron in culture infected with a GFP-containing herpes virus. And you can see the particles in the cell body, and you can see them moving out the neurites. These are individual virus particles that are moving in the processes of the nerve cell. And again, this can be done in culture or in animals, so the possibilities are enormous. We still detect DNAs by southern blot hybridization, where DNAs are fractionated on a gel, transferred to nitrocellulose, and then hybridized with specific probes. But this has given way in recent years to PCR, polymerase chain reaction, where we use specific primers to anneal to specific regions of a DNA and amplify specific regions. And the nature of the PCR reaction is such that after a few cycles of denaturation, annealing of the primers, and elongation to make the products, you get exponential amplification of whatever you're looking for. So here, the short product is flanked by the two red primers, and you can start with a very, very small amount of RNA or DNA and have enough to see on a gel. And this is how many, many infections are now diagnosed and detected. Here's an example of looking for this virus XMRV in chronic fatigue patients. So this was a paper from a paper that came out in Science last year. Chronic fatigue is a disease that's been around for many years, but which has no known etiology as of yet. And this group published a paper where they looked at 160 some odd patients with chronic fatigue and normal controls. And they had a reason to believe that this virus was involved in it. And they did PCR using probes specific for XMRV genome. And you can see a good number of the patient's sera were positive for one of the viral sequences. And normal controls, for the most part, didn't have the virus, but there were some. There's about 3 to 4% of their normal controls that have the virus as well. So that doesn't solve any problems, but it strongly suggested that this virus might be involved in the disease. And the other day I mentioned how it's possible now to discover viruses in the ocean, in Antarctica, in any environment, by a technique of high throughput pyrosequencing. And this allows you to do millions and millions of bases in a short period. You take a DNA that you want to sequence, you break it up and denature it, and you attach it to a very small bead. Then you do PCR to amplify the DNA on the bead. Then you put each bead in its own well in a very complicated automated apparatus. And then you do a reaction which is just brilliant. So when you have a single strand of DNA, if you add DNA polymerase and triphosphates, you will of course make a second strand of DNA. If you add one triphosphate at a time, you can tell which base is next in line to be polymerized. So what these machines do is very quickly put all 4 triphosphates in sequence in steps, and then they measure which one is incorporated. And this is possible because we have microfluidics. We have very small tubes that can carry amazingly small volumes very quickly. So let's say in this case there's a T about to be read next. The A will be polymerized there by the polymerase. You release pyrophosphate when you add the triphosphate. The pyrophosphate is converted to ATP by an enzyme that's included in the well. And that activates luciferase, which gives out light. So when you get a peak of light, you know that that's an A that should be there. So the machine tells you the sequence very quickly. It is, you can do millions and millions of bases. In fact, 400 to 600 million bases per 10-hour run. And this is why you can do the human genome in a week now. And you can easily take samples, total RNA samples from patients and sequence the whole thing and find small virus particles, small amounts of virus genomes in them. So this makes it now important to go back to the Koch's postulates that we talked about last time. I didn't really go through these, but Koch in the 1800s was the first to say how you should associate virus infections with disease. And he wrote up these 4 postulates. The organism must be associated regularly with the disease. You have to isolate it from the host and grow it in culture. You have to put the culture back into a host and reproduce the disease, and then re-isolate the organism. Well, these were great back then, but they don't work anymore. Not all viruses cause the same disease, if any. You can't often grow them in culture. You often don't have an animal model. So these are not practical anymore, especially when we identify pathogens by sequencing in PCR. And a number of viruses have been proven to cause disease long before they could be grown in culture. Hepatitis C virus, Kaposi's, sarcoma virus, and the papilloma virus is associated with cervical cancer. So we have to revise these postulates based on our new ways of identifying viruses. So here's just an example of a study where they looked at kids with respiratory infection. They looked for all the known rhinoviruses. There were about 100 of them. And a number of them, 52%, of these kids' samples didn't have any known rhinoviruses. So they amplified the sequences by PCR and sequenced them, and they turned out to be all brand new rhinoviruses. And they weren't identified before because they don't grow in cell culture. So we need to have new criteria for identifying pathogens. We need to have Koch's postulates for the 21st century, and they all involve nucleic acids. So we're talking now about sequences. A sequence should be present in most cases of a disease, should be in the places where the disease occurs. It should be few copies of the pathogen without disease. Now, the methods are so sensitive that sometimes you pick up pathogens even though someone is not sick. So you have to account for that in your postulates. When the disease goes away, the sequences should go down as well. When sequence detection predates disease, the association is more likely to be causal. That makes sense. The nature of the organism should be consistent with the biological characteristic of the group, and you should get tissue correlates at the cellular level. In other words, take tissues from infected people and show that there are sequences in the diseased tissue. And all of this should be reproducible. So that's not easy to fulfill all of these. And so that is why you will see in the literature now, XMRV, for example, there have only been a few studies done, and no one can really conclude whether that's the causative agent of this disease. Now let me leave you with 2 thoughts before we go. Remember, we've been talking about infecting cells in culture, where everything is reproducible and controlled. But in the real world, a virus has to find a host. It's not thrown onto a plate of cells. It can't get around. Viruses don't have locomotion, so it's not easy for viruses to encounter cells. The environment is very tough. Sunlight will kill many virus particles, drying, dilution, low pH. And once the host is encountered, the virus has to evade immune defenses, physical defenses, innate immune defenses, adaptive immune defenses. And all of this is not easy, but this is what we will consider when we talk about infection of a host. For a number of weeks now, we're going to consider virus replication in cells, but I want you to keep in mind that in a host, similar things happen, but the obstacles are much greater. We don't have nice homogeneous cells in a culture dish in a very nice incubator. We have different topological surfaces. We have different mixtures of cells. So all infections begin with a single cell, and all the things we're going to learn happen in the body, but it's very much more complex than that.